All right, and number 1 is Paul Andrews.
Thank you. A basic principle of evolutionary medicine is that the disruption of evolved adaptations can cause harm. Many in the medical community believe that antidepressant medications can save lives by decreasing the mortality associated with depression, for instance, suicidal behavior and cardiovascular disease. However, nearly all the commonly prescribed antidepressant medications target the serotonin system, which is itself an evolved adaptation, and it's intimately integrated with many adaptive processes in the body. So, in many processes other than mood, so including digestion, reproduction, metabolism. So the use of antidepressant medications could degrade the functioning of many processes regulated by serotonin and increase mortality. To test this, we conducted a meta-analysis on the all-cause mortality effects of antidepressants, and this is part of our results that you can see here. In patients who have not been specifically selected for non-vascular problems, so this is the majority of people taking antidepressants, antidepressant use was associated with a 37% increased risk of death. In patients who had vascular problems, there was no net effect of antidepressants on mortality, and that's probably because these drugs often have anti-clotting properties which might be useful in the treatment of vascular diseases. So in the vascular sample, the benefits and costs may balance out. So we also have some interesting results on mortality by the class of antidepressant. We're presenting that in our poster, and so please come and see our poster for that. How much time do I have?
About 20 seconds.
Okay, so I have just want to put in just a plug for two of people in my lab who are also doing posters and flash talks. Zach Driscoll, who's done the first clinical study of an adaptationist hypothesis.
That's going to be it.
Okay, thank you. Cheers.
Terrific. Thank you, and thanks for the time. Okay, next speaker, Veronica Berrigan.
Okay. Understanding leptospirosis is difficult. Leptospirosis is an infectious disease distributed worldwide, and it's caused by leptospira. That beautiful snow. It's me? Okay. I just have one slide. So that's leptospira, is a beautiful spiral bacteria. Leptospirosis is zoonotic with a wide variety of carrier animals, mainly mammals, as cows, dogs, pigs. When urine is excreted, large amount of leptospira are shed to the environment, exposing humans to the disease. Infection occurs when bacteria comes in contact with mucosal surface and skin abrasions. My research takes place in 2 low-income rural communities from Ecuador. It's a very small country in South America, from where many leptospirosis cases are reported every year. In order to understand better the transmission of leptospirosis in these localities, we need to look to all the potential sources of infection. But detection of the pathogenic leptospira by culture and by molecular methods is difficult. So I focus on developing a new test that detects very low amounts of leptospira in clinical samples and in the environmental samples. In urban areas, rats are well known to be the primary carriers of leptospirosis. However, in our study in rural communities, our results were different. Only 3% of rats were infected, in contrast with 20% of cows and pigs, and also 17% of environmental samples. Eradication of leptospirosis in this locality is a real challenge. We cannot kill cows and pigs as we do with rats, and we cannot throw antibiotics in salt and water. Therefore, better understanding of the ecology and evolution of Leptospira is crucial for preventing the disease.
Terrific, thanks. Jennifer Briner.
Okay, I'm Jennifer Briner, and this is Strategic Reproductive Effort by Life History and Drosophila melanogaster. And before I launch into this, I'd like to throw something out with regards to the Crespi-Badcock hypothesis. I was wondering if any of you guys thought of looking at fetuses conceived by donated eggs who have no relationship to the mother, and if not, that would be cool. So here I'm looking at trade-offs and compromises in Drosophila melanogaster. Because resources are finite, you expect to see a shift in how resources are allocated over a lifetime. So you can imagine with a very young organism, it would want to allocate more functions to growth than reproduction. But because over a lifetime, the odds of dying increase, there is less lifetime, you'd expect to see a shift in resources towards reproduction, and organisms should become more opportunistic as the odds of death increase. So we looked at 2 different populations, 5 replicates of each, and compared them in their reproductive ability, their ability to secure mates and secure territory over time, and that's it.
Thank you. Even with that, we kept to time. Don't forget to mention when you want the next slide. Next, Samantha Davis.
Hello, I'm here to talk to you about the Hispanic health paradox, which for those of you who don't know is this phenomenon in US health statistics where Hispanic groups have shockingly good mortality rates in comparison to the non-Hispanic population, which you would of course not expect given the socioeconomic difference. And we make, I think, in general, a lot of kind of weird mistakes when we discuss this phenomenon, one of which being that, of course, Hispanic is not one group. It's many, many groups. And, but the Mexican population is such a large majority in the US that almost all results in this phenomenon are really only applicable to the Mexican population, and all the other populations just sort of get subsumed within that. The second big mistake I think we make is that there are a lot of really major health behavior differences between the Mexican population and the non-Hispanic white population that, one, make the white population probably not a very good comparative population, but in addition are having large impacts on all of these diseases, namely smoking, but also obesity and alcohol behaviors and healthcare utilization. So what I attempted to do here was control for as many of those behaviors as was conceivably possible using one of the big national health surveys, and attempted to see if there was any leftover differences in risk, both ethnically and racially, and to compare against more populations than just the white Mexican population, which is what you're seeing over here. And I get what I think is a really interesting pattern that I think might help to explain some global differences in health statistics, and is probably related to some of the inflammation versus infection dichotomy that we saw in some of the earlier talks today. Am I right on time? Awesome.
Thanks. Perfect. Zachary Driscoll.
Thank you. So hi everybody, I'm Zach Driscoll. My poster is about some work that I've done at the Center for Addiction and Mental Health in Toronto in a sample of psychiatric patients studying the adaptive value of depression. So what we did was we directly tested the analytical rumination hypothesis, which if you're not familiar states that the symptoms of depression have evolved to promote prolonged focused bouts of analysis on one's problems and presumably the triggering problems for the episode of depression. And what we found was a strong, basically all our results support that hypothesis. We first found this correlation where the more severe depressive symptoms people have, the more analytical rumination they're doing with a questionnaire that we've devised and is well validated. And then we did a series of mediation analyses and model comparisons, all of which support this causal pathway, wherein the patients are perceiving their problems to be complex. This is triggering their depressive symptoms, and then those in turn are promoting those analytical ruminations. So our conclusions thus far are that clinical depressions may be caused by an evolved adaptation for coping with complex problems. And this, as Paul was saying, may have huge implications for how we treat clinical depression. Thank you.
So next time I think we should do this as a drinking game, and there should be like, no, seriously, there could be code words like every time someone said evolved adaptation, you. Just a thought. Okay, Julia Hyman.
Thank you. Our lab has been interested in looking at the effects of oxytocin on postpartum, both in healthy and depressed samples of women, but this is focusing on healthy women. So what we did in this particular study was we brought in, do I advance it? We brought in 29 women who were postpartum and 30 women who were nulliparous, and we compared them to a set of stimuli which had reproductive value in one sense or another. It included neutral, it included infants smiling, it included infants crying, it included sexual slides. Looking in this particular case specifically only at visual stimuli, because visual stimuli are particularly important in the early days, in the early months of raising an infant, when other forms of communication, particularly in humans, or other forms of communication are less accessible for affective learning. In this what we found over this is we looked at two areas. One is the amygdala, which processes emotional stimuli, particularly sensitive to emotional stimuli, usually negative, and these are actually positive stimuli. And what we found is that for sexual, neutral, and infant stimuli, the postpartum women had less responsiveness in the amygdala, in the right amygdala. So there's overall a decreased responsiveness in the amygdala. However, in looking at the VTA, the ventral tegmental area, which is around motivation and positive stimuli, rewarding stimuli, what we found there is we found no effect of cohort, meaning nulliparous versus postpartum, but we did find a strong effect of having given women on a double-blind basis nasal oxytocin. So they under the conditions of oxytocin, both samples of women had greater reactivity in the reward areas to infant and sexually crying stimuli. And I'll leave you with that surprising finding. Thank you.
Thank you. Michael Hoffman.
Teaching students and residents about frontal lobe syndromes is very, very perplexing. They may be completely silent, people may deny them, such as anosognosia, or people may have dramatic presentations, such as imitation behavior. Now, 34 million years ago, dramatic change happened on the planet. We went from an equatorial circulation of oceans to a meridional one, and Antarctic glaciation ensued, which caused global cooling, and that can be seen as the first part of the evolution of the human frontal lobes, a major initiating event. And therefore, not only is brain size a factor, but much more so is the neuropil enlargement. That's the most major difference between us and our closest relatives. Also, clinically, we find that looking at the human connectome and looking at connectivity in the brain is a much better way of analyzing frontal lobe syndromes. So this is a course proposal whereby I've been able to come up with 6 major groups, and I showed you the major increases in the arcuate fasciculus and the uncinate fasciculus in the previous slide. But there's other ones, for example, the frontopontocerebellar circuitry, whereby a cerebellar or brainstem lesion can present with a frontal syndrome, as they often do. So this evolutionary approach, the engineering approach to looking at frontal lobes in a connectivity way, is what the course proposal is all about. Thank you.
Jonjer. Go ahead.
Thank you. Could I have the slide, please? Or do I do it? There we go. The woman on the left has severe chronic pelvic pain and pain sensitization. If you take a Q-tip and run it down her tummy, she will demonstrate pain or allodynia in the region that's outlined. If you repeat it, it will move up segments into her abdomen. This is pain sensitization. It's a very conserved process. It's been identified in sea slugs that have not evolved at all. Interest in this has come about in chronic pain clinics where there's a huge representation of women with severe dysmenorrhea or severely painful periods. Severely painful periods or endometriosis produces this when the pain goes from cyclic pain to persistent pain. The woman on the left can't wear tight jeans. That's probably what bothers her most of all. The woman on the right has been treated with an evolutionary approach. She's had her menstrual cycle suppressed. Menstrual suppression reverses the pain sensitization. Thank you.
Next up is Laura Klein.
Thank you. Next slide, please. In this study, we examined the composition of 3 innate and 3 adaptive immune proteins in mother's milk from 7 populations. And as Katie Hine described earlier in her talk, these populations were characterized as having one of 4 subsistence patterns: horticulturist foragers, pastoralists, rural communities with access to shops and markets, or Western urban industrialists. The bottom of this slide shows simplified principal components analysis plots, where the shaded areas encompass the data points for all of the subsistence patterns, where the color scheme matches the map above. We found that the innate immune protein composition was more overlapping among populations, while the adaptive immune protein composition was more variable among populations and much more specific to subsistence pattern. Shared subsistence patterns likely share aspects of disease ecology, despite being geographically distant, though we still need to do more research to understand exactly what aspects of the local environment shape the immunological protection and instruction provided by mother's milk.
And people said that I was going to have to be mean. This is impressive. This is Harvey Kleiman. I may have to be mean.
I don't think so. The father wants the biggest baby possible. The mother does not want to die in childbirth. That conflict exists between the uterus and the placenta, and the battleground is the decidua. What we discovered is a technique that the father uses, the paternally controlled genes of the placenta use to immunologically divert the mother's immune system. Here is the normal situation where trophoblasts invade from the placenta into the maternal tissue, find the maternal spiral arteries, and convert those vessels. The mother would like to destroy these invasive trophoblasts. What the father does is secrete a galectin called galectin 13 or PP13. It goes out through the veins. It leaks out of the veins and precipitates, and this galectin is a master immune regulator, and it attracts the mother's immune cells to that region, away from the invasive trophoblasts. In simple terms, if we wanted to make some money for this new organization, and we want to rob a bank, before we do it, we're going to blow up the grocery store a few blocks away. While the police are in the grocery store, we go into the bank. That's what we discovered in the human placenta.
Shubhankar Kulkarni.
Yeah. Firstly, thank you for including flash talks in the conference. It's something new. So my poster is about my ongoing work as a grad student. And can I have the next slide, please? Okay. Contrary to the traditional view, which assumes that type 2 diabetes is a pathological departure from the normal healthy state, an emerging view depicts this insulin-resistant state as an alternatively evolved state fine-tuned to behavioral adaptations. So what we did is we compiled all available data from literature which on signals that affect insulin production, insulin resistance or sensitivity, and plasma glucose, and constructed a network out of these signals with their interactions amongst each other. Okay. Then we tried making all all sorts of perturbations to this network, and we found that no matter what perturbation is, the system invariably settles in one out of the two stable states. One of these states corresponds to insulin sensitivity and it correlates, and the second state corresponds to insulin resistance and it correlates. Both these states are stabilized by multiple mechanisms. This robust bistability points to an evolved system adapted to alternative metabolic personalities rather than a homostatic system with a single controlled normal state and the pathological departure from it in the form of insulin resistance. Thank you.
Thank you. Melanie Martin.
So global health recommendations for breastfeeding women are to exclusively breastfeed for the first 6 months of life, to reduce infant morbidity risks, and to promote optimal infant health. And yet, globally, most breastfeeding women do not exclusively breastfeed for that long. They introduce complementary foods well before 6 months of life. And this is because, likely, exclusive breastfeeding is time-consuming and energetically demanding. So this suggests then that the benefits of reducing exclusive breastfeeding outweigh the risks to infants for many mothers. And this would then predict that mothers who introduce complementary foods relatively earlier will show evidence of some improved energetic condition. So I looked at this in Tsimane mothers who, again, are forager horticulturalists. They all intensively breastfeed. They're not undernourished, but they do have limited energy budgets. And I actually found the reverse case. So I found no evidence of improved energetic condition in mothers who introduce complementary foods relatively earlier, instead adjusting for maternal age and time postpartum in a mixed effects model. Across ages, mothers who introduced foods relatively earlier weighed less compared to the mothers who introduced foods relatively later, suggesting that it's really only the mothers in relatively better condition who can afford the energetic costs of prolonged exclusive breastfeeding. And therefore, mothers who are introducing foods earlier perhaps have lower milk supply, are in poorer condition, or maybe also their infants are in poorer condition, and they are supplementing their infant diets, not supplanting them by introducing complementary foods and decreasing infant breast milk intake. Thank you.
Okay, and you guys have been a remarkably attentive audience. Congratulations for that. And so our final flash speaker is Dan Kruger.
Apparently I am the audiovisual device. So, being male is the single largest demographic risk factor for early mortality in technologically advanced populations. And in the college-age population, those 18 to 25, men are 3 times as likely to die as women. Did everybody know about that? And this is something that, it's a sex difference that stems from the basic properties of sexual reproduction. So females invest more, men compete for female partners, and this competition leads to riskier both behavioral and physiological strategies. There's multiple levels of mechanisms involved: genetic, behavioral, physiological, social, but they're all unified in a life history framework. The sex difference is based on the intensity of male mating competition, and the more intense male competition, the greater the sex differences in mortality. You can see this across the lifespan, between individuals within populations, across populations, and across populations that are undergoing changes over time related to male competition. So if we were able to eliminate this excess male mortality, we would save 300,000 lives a year in the US alone. And if you're not a big fan of men, you think, oh, well, these are just disposable people. Just the economic impact of lost work productivity alone would amount to $4 billion annually, which would easily close the budget deficit. Thank you.